welcome back to Virology. Today we are going to talk about mechanisms of pathogenesis. What is pathogenesis? It is all the processes that lead to disease. It can be viral pathogenesis, bacterial pathogenesis, any kind of agent. We're going to talk about viral pathogenesis today. And so a pathogenic virus causes disease, a non-pathogenic virus does not. And as you will see today, this is often a collateral outcome of the parasitic nature of viruses, which means they take something from the host, they damage it, but the host also responds, and that's part of the disease as well. And I just want to emphasize, before we go further, that selection does not select for viruses with increased pathogenicity. It typically selects for viruses which reproduce better or which may spread better from host to host. But increased pathogenesis is not a direct selective property. It may be indirectly selected, but it makes no sense to think about viruses being selected for more pathogenesis unless perhaps they reproduce better, so they spread better to another host, and the reproduction at higher levels caused increased pathogenesis. So that's a collateral damage. But we'll come back to that more when we talk about evolution. The way we study pathogenesis is through animal models. We generally don't use humans. It's not ethical, although there are some human challenge studies with viruses, and we'll talk about those later. But for the most part, you can't infect people with a highly virulent virus, right? You can't do that, even if they would like you to. So we use animals, and we use many different kinds of animals, but the mouse is a big one because we can make lots of mice, and we can genetically manipulate them. And so here on this slide are just some different ways that we can make mouse models for virus infection. So there in the middle is our mouse. Sometimes a human virus can infect a mouse. Not always, but sometimes. Some influenza viruses can infect mice, but not all of them. So you're limited to studying those viruses that will infect mice. Or if you're interested in a particular virus and it doesn't infect mice, sometimes there's an animal version of it that infects mice, and so you can model the human disease based on that. So that's the right side of the slide. But on the left side is we can genetically alter mice to make them susceptible to infection. So we can modulate the immune response, because sometimes immune response is what is limiting infection. So we can put clonal T-cell receptors in, we can delete T cells or B cells or interferon and so forth. These are really artificial circumstances, but sometimes we have to do that. All right, then we can put human tissues into mice. It's human xenografts. You can put liver in, you can put bone marrow and so forth, and make humanized mice so that now those cells can be infected with viruses. And then we can modulate susceptibility or permissivity. So receptors, sometimes the mouse doesn't have a receptor, so we just put the receptor in. Sometimes we can put individual viral genes or even whole genomes in. So a lot of ways to make mouse models. And of course, there are other animals that we use besides mice, not as common. Guinea pigs, hamsters, ferrets, and of course, non-human primates. But they're extremely expensive, and one costs $30,000, a little macaque, $30,000, and they're really cute. So you can't, at the end of the infection, you have to sacrifice them. It's not something you want to do. And you end up doing one animal per virus, and N equals 1, which is just not useful in my opinion. So we made a mouse model here at Columbia many years ago for poliovirus. I came here in 1982 and started working on poliovirus. One of my first graduate students, Kathy Mendelson, she identified the cell receptor for poliovirus, transmembrane protein. And then a second student, Rebel Ren, made mice transgenic for the human polio receptor gene. So mice, if you infect them with poliovirus, you can put 10 to the 9th PFU in, nothing happens, because there's no receptor in mice. But all you do is put the receptor in the mouse germline, and now they get poliomyelitis. You can see this animal has a paralyzed hind limb. So this is the first transgenic animal model for a virus disease. And after this, many other people have done it, and most recently, of course, ACE2 transgenic mice to study SARS-CoV-2 infection. But this, I think, is one of my greatest contributions to science, because before this model, they used to test polio vaccines in non-human primates. They would sacrifice hundreds and hundreds of them to test the vaccine. Now they do it in mice. And so I've saved a lot of non-human primates. I feel bad about the mice, but I don't know what to do about that. A big part of pathogenesis is tissue tropism, where the virus reproduces. So we have words for that. We call, if a virus reproduces in your intestines, we call it enterotropic, neurotropic for the CNS, hepatotropic for the liver. And viruses can be very limited in their tropism, or they can be pantropic. They can reproduce in many tissues. And so a big part of virus research is to understand what controls this, right? Why are some viruses limited and others pantropic? Because maybe you can get ideas about controlling them. And some of the factors that control tropism include the receptor, of course. If there is no receptor in a tissue, the virus can't reproduce there. So tissue-specific distribution of receptors controls tropism. Permissivity of tissues, right? Remember what that word means and what happens inside the cell. Accessibility of cells. A lot of cells are not accessible, easy to get to. And of course, defenses. Many parts of us are immune privileged, remember, and viruses can reproduce easily in them. Or virus defenses can be modulated. So all of these factors play into whether a virus can reproduce in a certain tissue or not. Here's an example of a tropism determinant. In other words, something that regulates where a virus is going to reproduce. It's cleavage of the spike glycoprotein of the virus. We've talked a lot about spike glycoproteins. You should understand what they are. And here's the hemagglutinin of influenza virus. And it's shown here as a red line. It's a transmembrane protein, of course. And it has to be cleaved by proteases to liberate the N-terminus where the fusion peptide is. And that has to be cleaved to allow fusion when the virus gets in an endosome and the pH drops. The proteases that cleave the HA can regulate tropism of the virus. So here, when influenza viruses reproduce in the respiratory tract, the epithelial cells, when they're released from cells, there are proteases in the respiratory tract produced by specific cell types that will cleave the HA right there. And those proteases are only present in the respiratory tract, and that limits most influenza viruses to the respiratory tract of humans. Now, avian influenza viruses, H5N1 viruses that infect birds and cause mass lethality, and which we are worried about because when they get into people, they can be lethal. Their HA is cleaved by a protease called furin. It's a family of proteases, which are in all tissues. And so that virus has a broad tropism as a consequence. It can get out of the lung, it can migrate elsewhere, and there will be proteases in other tissues that will cleave the HA and allow the virus to infect. And in fact, the ability of furins to cleave the HA of H5N1 is due to a few basic amino acids that are around the cleavage site. So a few amino acid changes introduce basic amino acids, and now furins can cleave those HAs and give you broad tropism. So cleavage of HA can restrict tropism. When a human gets an H5N1 infection, it's often infecting many different organs, not just the respiratory tract. TMPRSS2 is another determinant of cleavage. This is the cell surface protease that cleaves SARS-CoV-2 spike protein on the surface of cells. It liberates two parts of the fusion of the spike protein and allows the fusion protein to be exposed, and the fusion can then occur at the cell surface. So two examples of proteases that can modulate tropism. Many others, of course, but those are good examples that have been well studied. So the first question is, insertion of a furin cleavage site in the HA allows influenza virus to infect many organs. This means that the blank of the virus has changed, and you can put in that blank: susceptibility, club cell tryptase, permissivity, tropism, or all of the above. So putting in the furin cleavage site in the H5N1 viruses does what? 21. Okay, we're ready to see what we have here. Okay, the answer is tropism, of course. The insertion of a furin cleavage site changes the tropism of the virus because it allows cleavage by proteases that are present in tissues outside of the respiratory tract. It's not susceptibility. That doesn't make sense to say the susceptibility of the virus. Club cell tryptase, no, it doesn't make sense either. Permissivity, the virus is not permissive, it's the cell, it's tropism. I've been talking about pathogenicity, which is really a qualitative term. It means the quality or the state of being pathogenic, the ability to produce disease. And virulence, on the other hand, is the quantified aspect of pathogenesis, right? It's the disease-producing power of an organism which we can measure. Pathogenesis, you don't measure. You say a virus is pathogenic or it's not. There's no quantification. But virulence is the quantification of pathogenicity. So that's the difference between the two. So let's talk about how we can measure this disease-producing power of a virus, virulence. So if you have a virulent virus, how do we measure it? So first of all, we have virulent viruses and attenuated viruses that do not cause disease or cause less disease. And we can quantitate that in many different ways. We can simply measure the amount of virus in a host organism, and that can be a reflection of virulence. We can measure mean time to death. So in animal models, this is often how it's measured. You infect mice, and you see how many days it takes to kill half of the mice, 50% endpoint. The appearance of signs, right? You can measure fever, you can measure weight loss. You can't do symptoms with a mouse because they can't tell you what they're feeling, but we can do signs, and fever, weight loss are some of them. Measurement of pathological lesions. Polio virus, for example, will cause destruction of neurons in the brain and spinal cord. You could look at those under the microscope and quantify them. HIV-1 destroys CD4 positive T-cells. You can measure that. You can get how many T-cells there are per milliliter, and below a certain amount, you have what's called AIDS. So many ways you can measure that. And I just want to remind you that many signs and symptoms of disease are caused by immune responses. We've already talked about that, but we will talk about it more today. And remember, symptoms are what you feel, maybe with your fear also. Signs are what others detect. So here are two examples of measuring viral virulence. On the left, we have measuring death. It's the number of survivors on the y-axis, and on the x-axis is simply the day post-infection. And this is with poliovirus infection of mice. We have two different serotypes of virus, type 1 and type 2, which are genetically distinct. And you can see that when you put virus in mice, type 1 kills none of them. And we're just looking at killing. It doesn't mean it's not reproducing. All we're doing is looking at killing. And for type 2, the red dots, you see by day 10, all of the mice have died. So that's a difference in virulence, right? It's very obvious. Type 2 is virulent and type 1 is not. And you could quantify it by saying how much virus causes half of the animals to die, for example, a 50% endpoint. On the right is a different kind of assay where we are measuring pathological lesions. In other words, you take a tissue section, you put it on a slide, you look it under a microscope, and a pathologist can say, yeah, these cells are damaged, this is the kind of damage that's happening, and they can give a number to it. So this is an experiment where we have used, again, mice, and we've inoculated them with different viruses, Japanese encephalitis virus, yellow fever virus, West Nile virus, Longot virus, and dengue virus. And we have looked at different parts of the central nervous system, the cerebellum, the brainstem, and the spinal cord. and made sections and looked at them and given a score to them in terms of how much cell damage there is in each one. You can see, and that's plotted on the y-axis as a relative neurovirulence score. Neurovirulence would be production of disease in the central nervous system. So you can see that Japanese encephalitis virus causes high scores everywhere. Yellow fever, a little less, especially in the spinal cord. West Nile, high in 2 places, but low in a third. Longot is different. It's really specific for the spinal cord. And dengue is apparently not neurovirulent at all. It does not cause disease of nervous tissue, because that's what we're looking at here. We're looking at sections and asking for differences in the cells caused by virus infection. So that's just 2 ways that you can measure viral virulence. There are many, many other ways. The key here, though, the thing you need to remember, that virulence is a relative property. It's influenced by so many things: the dose of virus, the route of infection, the species of animal you're using, the sex of the animal, the age and the susceptibility of the host. As a consequence, we can only measure this in the laboratory, and very precisely. We cannot compare virulence of different viruses. You cannot compare poliovirus and Ebola virus. How could you do that? The assay is completely different. You can't do it. So if you have different polioviruses like type 1 and 2, which we just looked at, sure, you can compare their virulence because they're both polioviruses. We can't compare polio and West Nile. It's a different assay. Now, maybe if you did West Nile intracerebrally, you could get an idea. But it's really quite a different virus, and I don't think that would be valid. So this is why measuring virulence in people is very, very difficult unless you can do a challenge study, and you will never be able to do a challenge study with a very virulent virus because that's unethical. You can't make people very ill. So many people do what we call observational studies in humans. So during the pandemic, people would say, there's so many people coming to the hospital for this variant of SARS-CoV-2, it must be more virulent. This is the ultimate wrong conclusion to make. Here's an example. Virulence depends on the route of inoculation. This is a virus, lymphocytic choriomeningitis virus, and arenavirus, an envelope virus, which is a virus that reproduces in rodents. Usually doesn't get into people unless you are immunosuppressed. So my advice to you is if you have a pet hamster and you get an organ transplant, you give the hamster to someone else because you can get LCMV from it. And it's happened. If transplant patients have died of LCMV from their hamster. But in immunocompetent people, there's no problem. Anyway, in mice, if you give the mouse 100,000 PFU intraperitoneally, which you put the needle right in the peritoneal cavity, the mice survive. There's nothing going on there. They're fine. You see that mouse is fine. But you give 1 PFU intracranially right into the brain, the mice die. So the route of inoculation makes a big difference. Here it's just remarkable how big of a difference it makes. That's a dead mouse, by the way, there on the lower panel, in case you didn't know. So here's an example of a flawed conclusion, one of thousands made during the COVID pandemic. Here's a paper out of Canada. Progressive increase in virulence of novel SARS-CoV-2 variants in Ontario, Canada. So what they did, they measured hospital admissions over time from the first wave where you had the original virus and then Alpha and Delta over time. And they said, we're getting more hospitalizations, so it must be more virulent. That's an observational study. You can't control anything. You can't say it's more virulent. What's missing? Well, it's a different time, so things might be different. It's a different patient population. They could be different ages, different immune statuses. You cannot just tell, and people are now more aware of the pandemic, so they're more likely to go to the hospital and so forth. You don't actually know if they went because of COVID or with it as a consequence of something else. So all these variables, and this is just wrong. It's not true that there was increase in virulence, and you'll see the opposite, that it decreases in virulence. People will tell you Omicron is mild. We still have 2,000 people a week dying in the US. Is that mild? Obviously, you can't make those conclusions. You would have to do, best we can do is experiments in animals, but for SARS-CoV-2, the animals are pretty bad models of what happens in humans. So my take is, what's changed is the immune status of the population, and that's modulating disease, perhaps. You know, if you're immune, you're going to have a less severe disease. So the only way you can address these kinds of questions are by studies in animals. Now, in the laboratory, we study virulence because we'd like to identify virus and host genes that control it. And we could then use that for interventions of some kind. And what we do is we make mutations in the viral genome. And we look for viruses that don't cause disease in a particular animal model. So let me give you an example of that. Here's a virus that we can study in mice. We have the wild-type virus, which is the starting virus, and it grows well in cells in culture. These are plaques. You put it in the brain of mice, it multiplies, and it causes disease of the brain, which means it's neurovirulent, right? Neurovirulence is causing disease in the central nervous system. Then we make mutations in the genome of this mouse, and we have a virus that we've identified which doesn't grow very well, right? Fewer plaques and they're kind of smaller. You put it in mice, it doesn't reproduce well, and it doesn't cause disease. This is not particularly interesting because whatever mutation we've made simply makes the virus reproduce poorly. It doesn't say anything specifically about disease. But the third example is what we'd like to study. We make a mutation in a gene that does not affect reproduction in cells in culture, yet when you put it in the mouse, it doesn't cause disease. So that's a gene that's needed for making disease in an animal. It's not just needed for reproduction and fitness, which is important, but what we want to know is, are there virulence genes that modulate the pathogenicity? So that's what virologists have done for many years now. And over time, we've identified 4 classes of genes that can modify the virulence of viruses. We have, as I showed you earlier, there are genes that affect replication in a global way, and they're not terribly interesting. There are some viruses where there are genes encoding toxins that can kill cells. I'll show you an example of that in a moment. The big one is the class of genes encoding modifiers of host defense mechanism, right? Every virus has to antagonize host defense, otherwise it's eliminated. And if you change those genes, then the virus can be eliminated, and so it loses its virulence. And then finally, gene products that enable the virus to spread from one tissue to another in a host. And the last 2 classes, typically those mutations don't affect reproduction in cells in culture because, well, first there's no immune response except for an innate immune response in cells in culture. And there is no spreading within an organism. We just have spreading within cultured cells. So they used to be called non-essential, but that's a bad name because they're not non-essential. They're only non-essential because you can't figure it out in cell culture, but they have a big role in the host organism. Now, the virulence controlling genes do not have to encode proteins. Most of them do, but there are examples of sequences in viral genomes that regulate virulence that are non-coding. Here's an example from poliovirus. Remember, poliovirus has a plus-stranded genome with a very long 5' untranslated region, 743 bases. It's highly structured. The RNA forms stems and loops. A single base change in this stem loop 5 in the Sabin vaccine strains, these are the oral attenuated strains. We'll talk more about them later. A single base change reduces the neurovirulence of these viruses. And there are 3 serotypes of poliovirus, 1, 2, and 3, and each has a single base change in this 5' non-coding region. And we studied this in my lab years ago, and here's what we found. We made 2 polioviruses that differ only at 1 base, in this case 472, which is the base in type 3 that is changed in the vaccine. The vaccine has a U, the wild-type virus has a C. So the rest of the virus genome was exactly the same, 1 base difference. Then we put these intracerebrally into transgenic mice. And on the top here is the lethal dose, 50%. How much virus kills half of the animals? So you see the vaccine U doesn't kill any of them. And the C now is 9,000 PFU will kill half of the mice. So that one base change has reduced the virulence of the virus, the neurovirulence. And on the bottom is a growth curve in the brain. You inoculate mice intracerebrally, then you take samples at different times post-infection. You do a plaque assay, and we're reporting the virus PFU per gram. So you can see the virus where the C grows, it reproduces, and the virus where the U is cleared. So one base reduces the fitness of the virus in the neural tissue, and therefore it loses its neurovirulence. Some of the viral gene products, the proteins, on the other hand, that we've identified that modify, that can be virulence factors or determinants. Here are the immune modulators. We have a lot of those. We have genes that antagonize intrinsic responses like apoptosis, autophagy, ApoBac3G. We talked about some of those viral proteins that antagonize these cell processes. And if you mutate them, then the virus can't antagonize these processes, so they will be cleared and the virus will be less virulent. There is a class of proteins called virokines. These are viral proteins that look like cytokines. Remember, cytokines we produce in response to virus infection. They have a variety of effects. They cause inflammation and inducing the immune response. The viral counterparts look very much like them. They bind to cytokine receptors, but they don't act. So they block the cytokine receptors basically and prevent the real cytokines from doing their job. Then we have viroceptors, which are soluble cytokine receptors that do the same thing. They bind to soluble cytokines and they prevent them from binding to cells. So those are virus antagonists of cytokines. And if we make mutations in the genes that encode them, we make the virus less virulent. We have complement binding proteins. Remember, complement is a big antiviral pathway. MHC1 and 2 are very important molecules to display viral antigens to T cells. And there are modifiers of that, which we can reveal as virulence determinants. And again, all these are not required for growth in cell culture because we don't have much of these responses going on there. Here's another example of a virulence gene. And this one encodes a chemokine receptor. So it's a what did I call it? A viroceptor. So this is a virus called gamma herpes virus 68. This is a herpes virus of mice. So here's an example where we use a murine herpes virus to get some insight into human herpes virus infections. So what we've done here is to delete a gene called M3. And the assay is survival here. So we're looking at survival of mice with increasing dose of virus. So the wild type virus in blue, you can see with increasing doses of virus, kills the mice, and a 50% survival point is about right there. That would be about 5 PFU, right? Now, if you delete the M3 gene, which is the green, now the 50% lethality is over 100 PFU. So you've attenuated the virulence of that virus by removing the M3 gene. And then when you put the M3 gene back into that virus, it's M3-MR, you restore the virulence. Whenever you remove a gene from a viral genome, sometimes you can make unanticipated changes elsewhere just by mistake. So you have to put the gene back and make sure you restore the wild type phenotype. So this is M3 gene encodes a chemokine receptor, a viroreceptor, and that's why the mice are protected when you delete that gene. I mentioned that some viruses produce toxins. This is very rare. It's not like bacteria where many of them produce toxins of various sorts. Very few viruses produce toxins, but one of them is made by a rotavirus. It is an enterotoxin. So rotaviruses give you vomiting and diarrhea. This is gastroenteritis. And in the case of rotaviruses, which can cause that, they make a protein called NSP4, which is secreted from infected cells. So here's an intestinal epithelial sheet, and it's infected with rotavirus, and NSP4 is made. And NSP4 blocks the activity of a sodium glucose co-transporter. In other words, it's a membrane channel that exchanges sodium and glucose, and it also induces calcium in the cell. And all that does is cause an imbalance in fluid retention. So fluid is no longer retained in these cells. It comes out, and that's diarrhea. So this protein by itself can cause diarrhea. If you feed it to animal models, they will get diarrhea. So that's an explanation of the diarrhea caused by rotaviruses. Next question is, which statement about viral virulence is wrong? A, it can be influenced by dose, route of infection, species, age, gender, and susceptibility of host. It can be quantitated by measurement of fever. Ebola virus is more virulent than human papillomavirus. It is the capacity of a virus to cause disease in a host. When comparing virulence, the assays must be the same. So what's the wrong answer there? All right, what do we have here? What's the wrong answer? Yeah, Ebola is more virulent than human papilloma. You can't compare different viruses because the assays in the animals are going to be different. The other one that got a lot of attention is it can be quantitated by measurement of fever. For sure, you can measure virulence by fever. In fact, virulence can be quantitated, measurement of fever. All right, so those are virus virulence determinants. What about the host? The host also makes a contribution because when you get infected, when you infect the population, everybody responds differently. So there are host determinants. What are they and how do we measure them? Here's an example of determinants of herpes simplex encephalitis. We haven't really talked about herpes viruses yet, but when you're infected with herpes simplex virus, you get a lifetime infection of your peripheral ganglia. So one way you can get this is you get virus from your parents, from their saliva. It causes a cold sore and then becomes latent in the trigeminal ganglia, which are on either side of your head here. And periodically the virus reactivates, multiplies, and makes another cold sore. But sometimes the virus goes the wrong way. Instead of going to your lip, it goes to your brain. And that causes encephalitis. And this is one case in about 250,000 people a year in the US. If you don't treat them, 70% mortality, but they are treatable. And there are two peaks of incidence. One is at 6 months to 3 years of age, which would correspond to your first infection from someone else. And then later in life, reactivation from latency. So why is this so rare? What's with those people who get it and most others do not? So the way this is looked at is to take people who have had herpes encephalitis and who have not and do their genome sequence and look for single nucleotide changes in genes that associate with encephalitis. Now, our genome is 3 billion bases. That's a lot, right? You're going to get a lot of differences because every cell in your body has a different DNA sequence. So can you imagine comparing people? But sometimes you get very strong signals, like you get deletions that are very obvious. So this was done for herpes simplex encephalitis. And that whole procedure is called genome-wide association, GWAS. You look for SNPs in patients. So we call them SNPs, single nucleotide polymorphisms. They're not mutations. Who's to say that you're a mutant and someone else is not? They're just variations. And like John Steinbeck said, there's no good and there's no bad. There's just things that people do. There's no mutations, they're just SNPs. And so they identified for herpes simplex encephalitis changes in these genes, toll-like receptor 3, UNC93B, TRIF, and TRAF. These are all part of the TLR3 pathway. So TLR3 is one of the endosomal sensors of viral nucleic acid. And these other proteins are involved in the signaling from TLR3, like TRIF is one of the kinases that transduces a signal. UNC93B is involved in proper trafficking of TLR7. So all of these are involved with TLR3. And so basically these individuals have a reduced sensing and a reduced interferon response to herpes. They can't reduce the viral levels and it goes into their CNS. So higher levels of virus leads to CNS entry. So this is a very common way now of looking for host determinants. Sometimes you can find genes that affect susceptibility, which means the presence or absence of a receptor. And the best example is the CCR5 gene, which encodes one of the HIV receptors, right? There's CD4 and a chemokine, either CCR5 or CXCR4. And about 4 to 16% of people of European descent have a deletion in CCR5 gene and they don't make the protein. It's called CCR5 delta 32. This will protect you against HIV infection. So if you have this mutation, you're highly unlikely to get infected with HIV-1. And many years ago, there was a German AIDS patient who also had leukemia. He was given a bone marrow transplant to cure his leukemia. So they wipe out your bone marrow, and then they transplant into you from a compatible donor. The physician used a donor who was CCR5 delta 32, who did not have the CCR5 protein. So the physician was smart. He said, I'm going to do two things at once. This guy has AIDS and he has leukemia. So it cured him of AIDS. And then since subsequently, 4 other patients have been cured by this process. So it shows that CCR5 is essential. And this guy had tons of virus in him, but when he received a new bone marrow, all of his lymphocytes now were CCR5 delta 32. So that virus could no longer reproduce, and eventually it went away. Now you may say, why don't we use this to treat HIV/AIDS? Well, bone marrow transplants are really expensive and you can die. They have a very high complication rate associated with them. So they are not a solution to HIV infection. We'll talk more later about what might be. But this just shows you that changes in a receptor gene can affect infection. You may ask why 16% of people of European descent have this mutation, right? Why? I mean, I might. I've never. Actually, I know I don't have it because I did the 23andMe thing, and they will look for that, and I don't have this. My father is Italian, and so I would be a candidate, but I don't have it. Why, though? Something selected for it. Many years ago in the human population, there was some insult that killed a lot of people, and the survivors had this deletion, but we don't know what it is. There are lots of speculations in the literature, but we don't know what it is. So those are examples of host genetic determinants of virulence. We're going to get into some for COVID in a moment. But other determinants include age. And the example here is influenza virus. As you can see here, this is a map of influenza virus in the US between 1911 and 1915, so just before the pandemic of 1918. And you can see that the specific death rate was very high in young people, and then it became low. And then in older people, it went up again. So why is this? Well, we think, so very young and very old humans can be most susceptible to disease. Not always the younger ones, it depends. But in this case, we think the young are susceptible because they don't have a good immune response, perhaps, and they can't deal with infection. And then the older people, well, this is a respiratory infection. Their alveoli are less elastic, and therefore, if they're compromised by infection, they don't function well. They have weak respiratory muscles, so they can't compensate for virus in the lung, diminished cough reflex. They don't produce immune cells as well. In fact, as you age, your bone marrow diminishes, right? It gets smaller and smaller, so you have a lot of problems making new memory responses, particularly B cells from the bone marrow. I like to say I'm a shell of my former self because my long bones have very little marrow in them, but I still have memory to previous responses, but making memory to new one is very different. So here's an example of age effect on the severity of COVID-19. This is COVID-19 case fatality ratio. Remember, we defined this a little bit time ago. And this is by age. In just a few countries, South Korea, Spain, China, and Italy. And you see it stratified by different ages. So, and the case fatality ratio are the colored bars. So you can see at young ages, the CFR is very low. But then with increasing age, it starts to get higher and higher until it's the highest at 80 plus years of age, where in Italy, which has a very large senior population, it was over 20%. South Korea, Spain, and China. So age is another risk factor for severe disease. And we're not really sure. We have some hints, as I'm going to tell you here. So just to give you a perspective, if you're an unvaccinated person, you get infected with SARS-CoV-2, 40% are asymptomatic infections. Another 40% are pretty mild upper respiratory tract diseases, like you would call a cold. And then 20% of the people get pneumonia, where the virus is down in the lungs and you have a big inflammatory response, which is causing damage. And 10% of those are severe and require ICU admission and intubation and can lead to death. And about 1% of all these infections lead to death. But the risk of death, as you saw in the previous slide, increases with age. In fact, it doubles every 5 years from childhood onwards. There's also a sex effect. Men are 1.5 times more likely to die than women. So what's responsible for this age-dependent increase in disease? Well, a big factor is making autoantibodies to interferon. People over 70 years of age, about 4% of the population make antibodies to their own interferon. Of course, that's not good because you're blocking their ability to respond to an infection. And as you get younger, that percentage goes further and further down. And so I think that's shown in this slide here on the bottom here. So we have the red dots are autoantibodies to interferon. So you can see children and adults. These are just individuals trying to give you an idea of the fraction of the population. Very little autoantibodies in children and adults, but a good fraction of severe COVID can be explained by more anti-interferon antibodies in older individuals. And of course, most of the risk is unknown. Those are the gray dots here. We have no idea why they have increased risk. And that's also shown on the graph here. Autoantibodies to interferon going up with older age, and that corresponds with an increase in the infection fatality ratio, which is a better measure of death than the CFR. And the IFR is this black line, which you can see goes up here, and it's corresponding very nicely later in age to these autoantibodies. We also have what are called inborn errors of immunity. So these are mutations in genes regulating different aspects of the immune response. It could be interferon, it can be adaptive immunity. Those tend to be more responsible at a younger age, as you can see here. So in children, there's one blue dot for inborn errors, but that's a big fraction of the childhood population there compared to older individuals. So the line is going down there. And also people have done GWAS to say people who get severe COVID, what's the difference in their genome sequence from people who don't get severe COVID? And interestingly, there's a locus of genes that we have inherited from Neanderthals because we mated with them, right? And we retain some genes from them because they're apparently beneficial, but there are a cluster of genes that associate with more severe COVID that we got from Neanderthals. So it's the punishment we get for wiping them out, I guess. And then there are 5 other regions identified by GWAS that have changes that associate with more severe disease, and many of them are involved in interferon type immunity. So this doesn't explain all severe COVID, but it's a beginning, right? The age is a big one, and the antibodies to interferon. There's also a difference in sex response to infection. In general, men are more susceptible to infections than women, but not always. There's some exceptions to that. And here is an example of that. This graph is measuring neutralizing antibodies in people who get flu vaccines. So this is perfect. Everybody gets flu vaccine. So then you could do a study, take blood from men and women of different ages and compare the levels of neutralizing antibodies in the blood. So you can see y-axis, neutralizing antibody, seroconversion rate. Adult males have a lower antibody rate than do adult females. But aged males and aged females are more similar. So aged meaning beyond reproductive time, right? So the idea here is that, and this is actually phylogenetically conserved. It doesn't happen just in people. So the idea is it's important for reproductive success. So you protect the woman who is going to give rise, the female who's going to give rise to the offspring throughout the reproductive age. And then at an older age where you don't need it anymore, it declines. And this corresponds, this higher antibody level corresponds with elevated estradiol. However, I said this is not always the case. So in pregnancy, hepatitis A, B, and E, influenza, and COVID are all more lethal in pregnant women than in non-pregnant women or in men. Other determinants, malnutrition. This is why measles is 300 times more lethal in developing countries than in Europe and North America. Cigarette smoking increases susceptibility to respiratory infections. Air pollution increases respiratory disease, and stress. If you get stressed, you're more likely to have some kind of infection, including a reactivation of a herpes virus infection. Which statement about determinants of viral virulence is incorrect? Virulence genes can encode viral proteins. Virulence genes can encode cellular proteins. They are the same in all viruses. They can be found in untranslated regions. They may encode immune modulators. Oh. It's one person who did it. I think it's one person. Yeah, they're the same. Virulence is not the same in all viruses, right? Determinants of virulence are not the same in all viruses. So the one wrong answer, virulence genes can encode cellular proteins. Yeah, can, right? That's all these GWAS things, that's what they're identifying. So. But most of you got it right, it's good. All right, so how do virus infections damage the host? So there are two components. There's the direct effect of virus reproduction on cells, and I call that mechanisms of cell injury by viruses. So you infect cells in culture and they die. That's cell injury. So you can imagine if that happens in a host, you're going to get disease. So many viruses cause what we call cytopathic effects, right? They lyse cells. We looked at these a long time ago. Here's a time course of cells being lysed by a virus. They can cause syncytium formation, which we talked about earlier. The cells are fusing. Para-influenza, HIV, and others can cause cell fusion. That's part of the cell damage. Virus cells respond to infection by initiating programmed cell death. There are different kinds of that, apoptosis, necrosis, and pyroptosis. Many viruses encode what are called viroporins. These are proteins that insert into the plasma membrane, and then the contents of the cell leak out, so the cell eventually dies. Viruses can inhibit the macromolecular processes of the host, like protein synthesis, RNA synthesis, and this causes the cell to eventually break down. So many reasons why cells can damage, viruses can damage cells directly, but the more important cause of disease, tissue damage, signs and symptoms is the immune response, and we call that immunopathology. Fever, tissue damage, aches, pains, nausea, mental issues, cognitive issues, motor issues, may all be a consequence of the host response to infection. And especially for viruses that don't kill cells, non-cytopathic viruses, right? They're not killing the cell, they're not breaking it up. So what's causing the disease? It's the immune response. So to hammer this home, let's look at some examples. And here are some viruses and the different kinds of immunopathologies that they cause. And this is just a selection of them, but there are different categories. There's T-cell mediated. There are CD8 positive T-cell mediated immunopathologies. And those would be cytotoxic T lymphocytes that are killing infected cells. Then the CD4s, as you know, come in many types, including Th1 and Th2. There are immunopathologies due to both kinds of CD4 cells. There are even B-cell mediated immunopathologies due to antibodies. And we'll talk about some examples of these. But there's also over-exuberant innate immune responses. So much of severe COVID is a consequence of an over-exuberant innate response, which causes too much inflammation, and that's where you get a lot of the lung damage. In fact, when people are hospitalized, typically, that they are hospitalized because their blood oxygen has dropped and they can no longer function properly. They need to be intubated and given oxygen. At that point, the virus reproduction is already going down. It's the inflammatory response of the host that's causing that deficiency in oxygen. So let's take a look at some of these immunopathologies. So here's an example using lymphocytic choriomeningitis virus of mice, which if you infect mice intracerebrally, within 8 days, they're dead. Remember, this is the one where you put one PFU in the brain, the mouse dies. But if you infect and then immunosuppress, you give them a steroid, dexamethasone, to suppress broadly all the immune responses, the mice live. They have a persistent infection. You can't clear the virus when you're immunosuppressed, but the mice live. And then if you give them CD8 positive T cells from another mouse that's been immunized, it's called adoptive transfer or adoptive immunization, then the mice die. So the CD8 positive CTLs are killing the animals during the infection. So that's a beautiful example of immunopathology. These are some examples of what LCMV does and the mechanism by which CD8s are causing disease. So on the left, we're looking at mice lacking the perforin gene. So remember, perforin is one of the two proteins delivered by CTLs to cells which kill the cell. So you can make mice lacking the perforin gene. And if you infect them with LCMV, you look at the percent survival, the perforin knockout mice all survive. Whereas the wild type mice that have perforin, they die. So CD8 T cells are killing the animals. It's a host response to virus-infected cells, which is lethal. On the right is another measure of virulence. It's the presence of a liver enzyme in the blood. So normally your liver enzymes should be in the liver. They should not be in your blood at certain levels. If they are, it's a sign of liver damage. So we use this in many virus infections that target the liver. So you can see the perforin knockout mice. This is GLDH, a particular liver enzyme. It stays low. The perforin wild type mice, it's elevated because the virus is damaging, not the virus, the CTLs are damaging the liver cells. Many heart transplants are necessitated because people get an infection with a virus and it damages the heart, and by the time they know it, it's too late. So here, one of these viruses is a virus related to poliovirus called Coxsackie virus, and there's a mouse model for damage caused by these viruses. And on the bottom are two sections of heart tissue from mice that have been infected with a Coxsackie virus. On the left, A, you can see extensive cell damage. It's just stained with a dye that measures cell damage caused by infection. So those are cardiomyocytes, and around them there's a lot of cell damage. On the right, we've infected perforin knockout mice, and they have a beautiful heart tissue. There's no damage. So again, the T cells are causing the problems. There are also lesions associated with CD4 positive lymphocytes. These cells make a lot of cytokines. Now, CD8s lyse virus-infected cells, but CD4s don't do that. But they make cytokines that recruit a lot of other cells to the infection. These include neutrophils, mononuclear cells that cause a lot of tissue damage by releasing proteases and cytokines and so forth. So CD4s can induce a lot of lesions. And a disease called herpes stromal keratitis is involving CD4 positive T cells. It's a very common cause of blindness in developed countries. Essentially, you have repeated infections of your eye, and eventually it becomes opaque, and you can't see any longer. This is caused by CD4 positive Th1 cells. A virus replicates in the corneal epithelium, the outer layer of cells in the cornea. So here's a section of the corneal epithelium here on the left. And that top layer of cells, the epithelium, that is where the virus replicates. But the inflammation that leads to the blindness is in the underlying stromal cells of the cornea. And the problem, what happens is these infected cells release cytokines, and the cytokines recruit CD4 positive T cells. Here's another section of an infected cornea, and you can see all these dark dots are CD4 positive T cells being recruited by the infection, and they in turn cause the damage and the opacity of the cornea. So these cells are not infected. These stromal cells below the epithelial surface are not infected. They are damaged by the cytokines released by CD4s and by other cells that are recruited by that. Many viruses cause poxes and rashes. This is a form of immunopathology, measles, smallpox, varicella zoster. And basically you have virus reproduction in focal areas of the skin, but then Th1 cells, CD4 Th1 cells go in there, macrophages, they produce cytokines, they increase capillary permeability, more T cells come in, and that gives you the little rashes. So without the immune response, you wouldn't have many of these rashes. And let me give you an example of a immunopathology mediated by antibodies. This is dengue fever. It's a flavivirus transmitted by Aedes aegypti mosquitoes. And there's a group, by the way, called dengue fever. It's a Cambodian rock group because they have a lot of dengue in Cambodia. And it's endemic in areas where Aedes aegypti are present. Many billions of people are at risk because these areas are highly populated. You get about 400 million infections a year, and it's second only to malaria among insect-borne viruses. This is the region where dengue is present. And again, it's the range of the Aedes aegypti mosquito. We had almost eradicated dengue from South America before 1981 by the use of DDT. Kill the mosquitoes, but we learned that it's not good, so we stopped using DDT, and dengue returned after 1981. And many of the mosquitoes were reintroduced by the tire trade, the recycling tire trade. You have old tires you throw onto ships, they fill with water, mosquitoes breed in them, and then you transport them all around the world, and you bring Aedes aegypti to different parts of the world. Now, the problem with dengue is that the first time you get it, it can be asymptomatic or an acute illness, headache, back and limb pain, rash, aches and pains in the bone. Not pretty, I understand, but you recover. But there are 4 serotypes of dengue virus. And if you get infected with one, you're only protected against that one serotype. You also have a complication, hemorrhagic fever, which is about 1 in 14,000 of primary infections. You get bleeding and shock syndrome. The key here is that these antibodies don't cross-protect. So if you get infected with type 1, you're only protected against type 1 and not types 2, 3, or 4. But let's say you get type 1 year, and then the next year you get infected with dengue type 2. You make a memory response to type 1. You make antibodies to type 1. These will bind the virus, but will not neutralize it. They will allow the virus to attach to FC receptors on macrophages and other cells that are not normally infected, so you have an increased burden of infection. And these cells release cytokines and chemokines, and that gives you this hemorrhagic fever and shock syndrome. So it goes from 1 in 14,000 in primary infections to 1 in about 90 or 1 in 50 in a secondary infection. So what's the solution? It's to vaccinate everyone. And now we have some newer dengue vaccines which seem to be able to protect against this. And finally, viruses can immunosuppress, another kind of pathology, global reduction of the immune response. The virus can reproduce in cells of the immune system. They can perturb the production of cytokines, and viral proteins, as we said, can inhibit these reactions by being viroceptors or virokines. And here are 3 examples of immunosuppression. Measles is a big one. It infects monocytes, dendritic cells, and causes enhanced infections. As you'll see, it does something else that's quite bad. Rubella reproduces in lymphoid cells, and of course HIV reproduces and destroys CD4 positive T cells. And so the consequence is you get infections with other pathogens when you are immunosuppressed by these viruses. And for HIV, the cause of death is by these opportunistic infections that occur because you no longer have an effective immune response. It turns out, a recently discovered phenomenon, when you are infected with measles virus, not only does it immunosuppress you, but it wipes out your immune memory. Remember, immune memory consists of B and T memory cells that last, right? Measles virus infects those cells and destroys them. So if you've had a bunch of infections as a kid and a bunch of vaccinations, if you get measles, it's all out the window. You have to get them over again. So please tell people who don't want to vaccinate their kids for measles that this is a severe consequence. This is the experiment that showed it. It's called VirScan. They take overlapping peptides covering 400 pathogenic viruses, all the proteins of 400 pathogenic viruses. They made individual peptides. They put them on a plastic surface, and then they take serum from kids with and without measles and run it over. So you can get an idea of the infection history of these kids. And if you just look, these are the dot plots. These are so many dots in here, each corresponding to a simple epitope that they just appear as a smear. But here are control kids who have never had measles and measles vaccinated kids. And here are kids with mild and severe measles before and after measles. You can see this dramatic drop in the number of peptides that they recognize. In other words, their immune memory is being wiped out. So it's immune amnesia. The measles eliminates between 11 and 73% of the antibody repertoire. So a good reason to vaccinate. And as you may know, right now in the US, we're having outbreaks of measles because parents think they shouldn't vaccinate their kids. They get religious exemptions so they can still go to school. Politicians in certain states think they'll get elected by saying you shouldn't get measles vaccination. So we're going to have a big measles outbreak in this country in the next few years. And these kids, besides the risk of encephalitis, which we'll see next time, and other consequences, they're going to have their immune memory wiped out. Good move, folks. Next time, we're going to look in detail at some specific acute infections.